Hello everyone, I thought I'd make a video for your lab this week. I'm having issues getting the Flash projects or Flash modules to show up. So I'm going to go down here and just have a real brief introduction of what you're going to be doing. So I'm going to click on this. Now, this is the sheet you're going to use. When you click on that, it's going to ask you to download it. So this is going to be your answer sheet. I already have it open here. And when you're done, this is my view, so do what you did last week. Please submit it that way. It works out a lot better. It's easier to grade that way. More reliable. I know I'm going to get it. So what you're going to be doing, this is based on what we're covering this week, is diffusion osmosis. There are a couple of sort of introductory animations you can watch. This is the first one. It's over diffusion. Pretty cool.
Molecules dissolved in a solution are in constant random motion due to their...
This happens to be the same ones in your lab lesson. Somehow I ended up picking the same one that Park has used for their online labs. And then this is one with osmosis. Now,
diffusion is the net movement of molecules.
Just clicking on it. So here's the membrane. Most of the time you're dealing with some kind of a dialysis type tubing thing that allows certain things to cross over and certain things cannot. So it's termed a selectively permeable membrane. Now, a lot of references will say that osmosis is diffusion of water molecules. That's true, that's correct. But osmosis always involves water. It does not involve solid particles, but is sometimes referred to as diffusion of water molecules. So what you're going to do, I'm going to go to the lab answer sheet. Here's a little bit of reading. You're going to be using dialysis tubing. Now, if you don't know what that is, dialysis tubing is used a lot of times in science experiments. This is dialysis tubing. It's actually designed as a selectively permeable membrane. It goes in dialysis machines. It's meant to be sort of act like a kidney glomerulus in the filtration process, but the way it works, it's a selectively permeable membrane. That means small particles, what will fit through the membrane pores, will actually move out of dialysis tubing. Larger molecules cannot, so they will stay inside. So it's pretty common in an experiment to use dialysis tubing when you're talking about movements across cell membranes. You see this example, they're going to pour in some, looks like molasses. But I just wanted you to see what you do is you tie the bottom of the bag, you pour something in, you tie the top of the bag, and then you submerge them in water. And that's what you're going to be doing, is interpreting what happened with that. So now that you've seen how you set up an experiment with a dialysis bag, what you're going to do, you're going to have to interpret an experiment. This term grams per mole, that's a mass number, a unit of size. How big is a substance, a particle? In your experiment, you're going to have to interpret. What you're going to do is a dialysis bag is filled with a mixture of glucose and protein. You saw how that happens just earlier, or how you can do that. You take a small subsample of the dialysis bag. You're also always going to take a sample of what's the bag being placed in. Now, look at this, 180 grams per mole. This is 10,000 grams per mole, so clearly glucose is a lot smaller molecule than protein. Grams per mole is kind of a standard unit. Sometimes it's kilograms per mole, but there's a setup. And here's the water, here's a dialysis bag, and it has glucose and protein inside. So this design of an experiment, what you do normally is you do something like this, and then you have to test your results. To test for glucose, Benedict's test is always really common. It's a blue chemical. It's got a really pretty blue color to it. Biuret test is the presence of proteins. That actually tests for peptides, which are what proteins are made of. If glucose is present, Benedict's will turn green. If there's no glucose, it stays the color of Benedict's, which is blue. If protein is present, the Biuret indicator will turn violet. So fuchsia. pinkish, maybe. Close anyway. If there's no protein, it remains clear. And like I said, you would test the dialysis bag and the water. So what ended up happening is you soak this bag for, I believe it indicates here, for 60 minutes. They then did some testing on the bag and the water. So here's the dialysis bag across, here's the beaker of water. So beginning green for glucose, because yes, you put it in there. In the end, there was still glucose in there as well. Beginning for protein, violet, because it was added, and at the end was violet. So that's in the dialysis bag. The beaker, you can see here, beginning was blue, so there's no glucose. At the end, it turned green. And then here it was clear at the beginning because there was no protein. And then here it's clear. So what you're going to do, I had to look at this a couple of times, but summarize the results regarding the presence or absence of glucose and protein in the table below. So you're going to put a plus or a minus. Okay, if you thought there was a positive result, like here, green in the dialysis bag, remember earlier it said if there was glucose present, that Benedict's would turn green. So that would be a plus. You'd put a plus in this box right there. Just like that. So fill this out. Give me your interpretation of what happened here as far as in this table. Explain the movement of, or lack of movement of protein or glucose. Why did one molecule move and the other didn't? What factors affect movement of molecules across a semi-permeable membrane? You got to think concentration gradients. You got to think about the size of the particles. Yeah, and just briefly explain active transport and how it differs from passive transport. So just two definitions, kind of a compare and contrast. How is one different than the other? There's one glaring major reason. We covered that tonight in class, and it's in your outlines in a PowerPoint. Okay, so that's the first part of your experiment. The second part involves osmosis. Now, you're going to define these terms. We went over this in class, and then you're going to go to this website. So I can I'm going to control click on that. Now, you can't, I wish you could, just going back here, it says the first thing I do is click on review. What I was going to say is I wish you could make this bigger, but you can't. So click on review, and even that little window pops up. See, if you make it larger, my computer's a little slow. It'll, there it goes. It still stays the same size. A little overview of the three, what are called the tonicity of fluids. So you click next. This is isotonic fluids. It asks you some questions based on that. Go through and what you're going to do is, wrong one. You're going to hit play, kind of gives you a little visual on what's going on with isotonic as far as movement of water molecules. You click next. This is hypotonic solutions, so you can click play. Gives you a little animation on that as well. What happens with hypotonic solutions that cells are sitting in. And then you're going to click on this. It gives you a little comparison between an animal cell and a plant cell. Now, every example that I've ever seen really with osmosis usually deals with red cells. They're pretty fragile, but human cells can change shape drastically depending on what kind of fluid they're sitting in. But you can hit play here. It shows you what happens with the plant cell. Hit play, shows you what happens with the human cell. Yeah, and red cells are pretty fragile, so they burst. And then it says back to review. And you just kind of shuffle through and do all. And there's another example with plants, this hypertonic solution. So go through, answer these questions. What concentration of salt is isotonic to animal cells? I'm going to go over that with you as well, or I did go over that with you, and then that's also in that little beginning paragraph. When cells in isotonic solution, is there movement of water in or out of the cell? So we will have covered all this. Go through the hypotonic solutions. Describe the net movement of water in a hypotonic solution. Why does water move this way? It's based on a property of water, so we're going to go through that. And then same thing here. Look at the plant cells. What happens to an animal cell when placed in a hypotonic solution? What happens to plant cells? Plants actually thrive for that. That's why you water plants. They're loving the hypotonic environments because that helps force water inside the cells. And the last one would be over hypertonic solution. So just follow these instructions. There at the end, you have a couple more questions, and there's the rubric. So when you're done, save your results, and we'll go from there and turn it in. Thanks.